Hi everyone, my name is Janet. I am joined today by my group members Abigail and Alexander, and today we'll be talking about the 2022 Nobel Prize in Physiology and Medicine. People sometimes use the word Neanderthal as an insult to suggest that a person is brutish, unintelligent, or rude. However, it is very possible some of you have a percentage of your genes inherited from the Neanderthals. You may even have genes from other ancient human species. How is this possible? Where do we come from, and how are we related to our ancestors before us? What makes us, Homo sapiens, different from other hominins? These are all questions Swedish scientist Savant Pabo sought to answer. His journey began in 1984, a time where recovering DNA from long-dead organisms seemed purely science-fictional. While working towards his PhD at Uppsala University, he attempted to sequence DNA from Egyptian mummies. Unfortunately, his samples were contaminated with modern human DNA, so his results were no good. He didn't stop here. In 1990, Pabo started his own research group at the University of Munich, where he was the first to successfully retrieve and sequence bits of ancient DNA from a Neanderthal in 1997. After refining his methods to avoid contamination, his team sequenced a complete Neanderthal genome in 2010. His hard work led to his 2022 Nobel Prize in Physiology and Medicine, which was awarded for his discoveries concerning the genomes of extinct hominins and human evolution.
The name of the paper chosen for the analysis and comparison of Neanderthal genome to the present-day genomes of Homo sapiens, commonly known as human beings, is titled A Draft Sequence of Neanderthal Genome and is written by Richard E. Green, Svante Pääbo, et al., published in April of 2010. The question Pääbo set out to answer was whether it was possible to extract DNA samples from Neanderthal fossils without contamination by modern human DNA, and whether modern sequencing tools could be applied to arrange fragmented DNA into a logical sequence. Once the sequence was obtained, the researchers expanded the scope of the project to determine the extent of overlap between Neanderthals and modern humans, and to identify specific sections of the genome where those differences exist. It also aimed to answer the question of whether interbreeding events had taken place between Neanderthals and modern humans, and whether that genetic exchange contributed to the genetic makeup of present-day humans. By understanding the genetic relationship between Neanderthals and modern humans, it is possible to reconstruct the evolutionary history of our species. Svante Pääbo and colleagues employed a paleogenomic approach to address the questions regarding the genetic relationships between Neanderthals and modern humans. The approach involved several key methods. Method number 1, DNA extraction from Neanderthal remains. Researchers isolated ancient DNA from well-preserved Neanderthal specimens, often obtained from archaeological sites. Method number 2, genomic sequencing, which involved sequencing the Neanderthal genome and drafting a sequence that represents the genetic makeup of Neanderthals. Method number 3 was the comparative genomic method, which involved Neanderthal genome compared to genomes of modern humans to identify similarities and differences. This involved analyzing genetic variations such as single nucleotide polymorphisms and structural variants across the genomes of Neanderthals and present-day humans. Method number 4, population genetic analysis, which involves statistical methods that were employed to assess levels of genetic divergence, population structure, and admixed between Neanderthals and modern humans. By combining these approaches, the researchers were able to reconstruct the genetic relationship between Neanderthals and modern humans, shedding light on evolutionary processes such as interbreeding and genetic adaptation throughout human history.
In the heart of Vindica Cave in Croatia lie the ancient remnants of Neanderthals waiting to reveal their secrets. Researchers used a sterile drill to remove 50 to 100 milligrams of bone powder from Neanderthal bone fossils for genomic analysis. However, unlocking the mysteries of the Neanderthal genome is far more complicated than simply collecting bone powder, and the researchers were faced with many unique challenges. Many DNA fragments in Neanderthal are identical to present-day humans. Therefore, the slightest contamination of experiments with modern human DNA could skew their findings. Sequencing the Neanderthal genome was once considered impossible due to the large proportion of microbial DNA in the specimens, as well as the highly degraded quality of the DNA samples, which were on average only 200 base pairs long. To circumvent these challenges, the researchers handled samples in a clean room facility and tagged them with a distinctive 5' TGAC 3' sequence to distinguish the DNA from modern genetic material contaminants. Once the DNA was extracted, it was then amplified and enriched using the precision of restriction enzymes to surgically excise microbial DNA and elevate the proportion of Neanderthal DNA within the sample. Samples were then sequenced using a next-generation sequencing platform to enable high-throughput sequencing of the millions of DNA fragments. Once the samples were prepped for next-generation sequencing, the next step is library preparation and sequencing. Genes were amplified by PCR, and the molecules were sequenced from both ends, though all reads required correct cleanroom tags. The bases were called using a machine learning software. These sequencing platforms aren't perfect, and nucleotide misincorporation is possible due to the deamination of cytosine residues. To account for potential misincorporation, the researchers aligned the sequences to the reference human genome HG18 and the reference chimp genome PanTro2 to determine an error rate of around 5%. It was unknown to the researchers whether the bones came from 1, 2, or 3 separate individuals. They analyzed the complete mitochondrial DNA sequences and determined that the 3 bones came from 3 different individuals. Next, they sought to determine the extent of contamination by modern human DNA. Because at the time of this study there was no known fixed difference between Neanderthal and present-day humans in the nuclear genome, the extent of contamination was determined by searching for the presence of Y chromosomal DNA in female Neanderthal samples. They determined the estimate of contamination was 0.7%.
By comparing Neanderthal genome with modern human genomes, the study revealed both similarities and differences, showing that Neanderthals and modern humans share ancestry, but have undergone genetic changes over time. What's really interesting is that the study provided strong evidence for interbreeding between Neanderthals and modern humans. Modern humans outside of Africa were found to have a small percentage of Neanderthal DNA, suggesting gene flow between the two groups when they lived together in Eurasia. This finding sheds a light on the population dynamics, migration patterns, and genetic diversity among Neanderthals and early modern humans. Additionally, the study identified parts of the genome where Neanderthal DNA is present in modern humans, revealing the genetic legacy of Neanderthals in today's populations and its impact on human biology, disease susceptibility, and adaptation. The results greatly enhance our understanding of the genetic relationships between Neanderthals and modern humans, providing a valuable insight into human evolution and population history. One key finding of the paper was to determine how much the Neanderthal genome diverges from the human genome in areas different from human chimpanzee common ancestor. They sequenced the genome of 5 individuals from different origins: French, Han, Papuan, Yoruban, and San, and compared them to the human reference genome HG18 in segments of 100 kilobase pairs long. They did the same for the 3 Neanderthal genomes shown in red, orange, and yellow. The average divergence from the reference genome was greater in the 3 Neanderthal genomes, with about 11% average divergence, while the present-day human genomes diverged by 8%.
By probing the areas of divergence, researchers could then identify specific genes that were present in humans but absent in Neanderthals.
One important implication of this research is the potential implications their findings present for the origins of modern humans. The genetic data supports two possible models that have led to modern humans we know today. The first model suggests gene flow occurred from Neanderthals to Homo sapiens as they left Africa. This is the preferred model since modern humans with European or Asian descent were found to have approximately 1 to 4% of the genome that originates from the Neanderthals, while humans of African descent do not have Neanderthal DNA. The second model states that African populations might have migrated and replaced hominid species without intermixing with Neanderthals. Though their evidence proved otherwise, this classic model still supports the idea that the vast majority of genetic variants that exist outside of Africa today came from Africa with the spread of modern humans. Svante Pääbo rightfully earned his Nobel Prize. He not only developed techniques to successfully sequence the Neanderthal genome, previously thought to be an impossible task, but his findings shed light on the evolutionary history of mankind and our hominin relatives. His evidence of gene flow from Neanderthals to modern humans and the analysis of the Neanderthal genome shows that they are likely to have had a role in the genetic ancestry of present-day humans outside of Africa. Future analysis of the Neanderthal genome as well as the genomes of other hominin species can fuel hypotheses and provide further insights into the origins and early history of present-day humans. Researchers could also see the functions of genes with similarity and see if there are any advantages or disadvantages of having Neanderthal DNA. Here are all our references used. We hope you enjoyed and thank you for watching.